Hi, everybody. Welcome back. In this video, we're going to introduce a cell fate decision, one of the simplest ones that really exists, a cell fate decision that's made in viral infections of bacteria, decision between lysis and lysogeny. This captures, although it's a simple system, captures many of the underlying features, genetic regulation, that play similar roles in more complicated cell fate decisions, for example, in people, but is simple enough that we can actually understand it, write down fairly simple models of it, and work with it computationally. This is the system that's going to form the core of the second MATLAB project in the course as well. So let me begin by switching over to the slides so that we can get started. So first of all, let me introduce the idea of phage or bacteriophage. Bacteriophage, quite simply, are viruses. And specifically, they are a type of virus that infect bacterial cells. Lambda phage is one of these. There are many different phage with various sorts of names associated with them, Greek letters, letter number combinations, and so on. Not particularly interesting names, if you will. And they all have a structure that is entirely made up of a relatively small number of proteins with a head unit, as shown here, that has DNA sort of trapped inside it, the DNA, the genome of the phage, and then some other proteins, for example, tail proteins and some fibers that help attach to the target cells. And the little piece of DNA, the genome that is inside this head, has the sequence for all of the proteins that make up this virus, as well as a few other proteins that play regulatory roles. And a couple of these we're going to discuss further on in this lecture. What usually happens, the so-called normal fate of bacterial infection, is highlighted here. This is a fate known as lysis. This graphic actually discusses a T4 phage rather than a lambda phage. Very, very similar viruses, very similar life cycles. First of all, the virus lands on the surface of the cell. This is not to scale. The virus is in fact much, much smaller than the cell. Then the virus injects the DNA into the host. And once that DNA is inside the host, it kind of takes over the machinery of the virus, or sorry, the machinery of the bacteria that normally is used for expressing proteins of the bacterial cell, and instead takes over that to make new copies of the virus of the viral DNA, as well as new copies of the proteins that make up the virus. These assemble, the heads assemble around the DNA, the spikes assemble and everything. And then finally, you end up with complete viruses, maybe 100, 200 new phage inside a cell, and they then cause the bursting or lysis of the cell. And that releases the phage out into the environment that then let them infect new cells. And so each infection leads to rapid production of new viruses, releasing 100 or 2 new viral particles out into the population. And this can rapidly make its way, basically destroying, killing off an entire colony of bacteria quite quickly. However, sometimes this doesn't happen. The exact frequency of how often it happens depends a little bit on which phage you're talking about, but it's a fairly common mechanism across phage. Sometimes something else happens. If you're looking through, say, looking at a population through the microscope, you might see many viral infections leading to lysis and the cells burst. But some bacteria seem to get infected. The virus lands on the surface. We can't really see the injection of the DNA, but if you could, that happens. But no phage are produced. The bacterial cell doesn't burst. These cells continue to grow and divide. They seem to be perfectly normal. And this can happen for generations and generations, the cell dividing and dividing and dividing. But they are in fact infected. And then we know that they're infected because under stressful conditions, if you say expose the cells to UV radiation or something like this, maybe 10 generations after the parent was, or the great-great-great-grandparent was exposed to a virus, these cells will all of a sudden starting producing new phage and lyse. The bacteria appear to have what you might call a latent infection. The virus is hiding inside the bacteria somehow, waiting for an opportunity to reproduce. This alternative fate is known as lysogeny, right? And this decision pathway is kind of captured here. We have our initial phage that lands on the surface of a cell, injects its DNA into the host, and then there's a decision factor, decision state. Either the cell undergoes the infected cell undergoes the lytic cycle, where new phage are produced and the cell lyses and releases new viral particles, or that viral DNA gets integrated. Actually, it gets sort of spliced into the DNA of the bacteria. And the bacteria just goes along, reproducing, living normally, perhaps many, many cell divisions, where each of these daughter cells, progenitor cells, have the prophage, the phage DNA sitting inside their genome. And occasionally, due to random chance, but more often triggered by a stress event, there can be a switch back into the lytic cycle. This captures really a lot of the fundamental ideas of a cell fate decision. This is absolutely a cell fate decision. We have a cell that is in some particular state, and a decision needs to be made as to whether the lytic cycle is entered or the lysogenic cycle is entered. Right? These are stable states that can, the lysogenic state can persist for generations. Right? Both of these states, in fact, are fairly stable. The lytic cycle, once it is begun, never transitions back. You never see a situation where a cell started to engage in the lytic cycle, produced a few copies of the virus, and then went back into lysogeny. This doesn't happen. Once the lytic cycle has clearly been selected, it completes until the cell is lysed. Similarly, once the lysogenic cycle is chosen, it tends to be stable. It can be stable for many, many, many generations. And if there's not stress in the environment, the transitions back to lysis are very rare. However, when there is stress, there actually can be quite a robust transition to lysis. Now, there's a number of questions that we can ask about this lysis-lysogeny pathway. One of the first of these is, what makes the decision happen? Why do some infections lead to lysis and some lead to lysogeny? Can we understand what is driving that decision. What causes the switch? Why is the lysogenic state generally stable, but when there's stress, it switches back into lysis? These are sort of questions of what or why. That is, how do these things happen? What's controlling these? A related question is one of prediction. Can we predict the outcome of a specific instance of infection? These are the same sorts of questions that we would want to ask about more complicated cell fate decisions, like those underlying development or blood cell differentiation inside adult mammals and so on. Can we understand what's causing it? Can we understand what causes transitions between states under that of stressful or non-standard environments? Can we predict what would happen under certain circumstances? And perhaps we might add a fourth option here. Can we interfere with this process in some way? Would it be possible for us to affect the outcome of a particular infection through some intervention? Right. Now, this is a system that has been very well studied, both experimentally, and actually there's a fairly rich literature on computational modeling of this system if you want to look that up. And I'm going to give a very kind of rough overview of this. The point of this isn't to get into nitty-gritty details of how things work, but a rough idea. Generally, the phage genome is structured by dividing it into two pieces with some control elements, that is, places where transcription factors can bind in the middle of them. And very roughly, one group of the two involve genes required for lysogeny, the genes that promote the lysogenic state, and the other group contains genes that are responsible for propagating lysis, the lytic pathway. Each of these groups contains a transcription factor. The group of genes responsible for lysogeny contains a protein referred to as CI. This is sometimes simply called the lambda repressor. CI forms dimers. It's a dimeric molecule. It's this complex of two molecules stuck together. And the genes for lysis also involve a transcription factor. This transcription factor is known as CRO. CRO also forms dimers. And in between these, there are some transcription factor binding sites. There's actually a little bit more richness than just the three shown here, but the three shown here are dominant and sufficient to capture the main ideas of what's going on. So putting this together, we can think of, we have this genetic structure where one half of the DNA has the potential to make a messenger RNA that codes for CI, a transcription factor, as well as the other mRNA for lysogenic genes. And of course, that mRNA can then be translated and then folded into a CI protein, which again typically forms a dimer, and as well as the other proteins responsible for lysogeny. The same thing can happen on the other side. The gene for transcription is able to be transcribed into messenger RNA for the transcription factor CRO, as well as the rest of the lytic genes that can then be translated into protein. Again, CRO tends to fold the dimer, as well as the other proteins responsible for lysis. This gives structure. It doesn't tell us anything about the mechanism of how this is controlled. Clearly, there's going to be an issue if both the lysogenic genes and the lytic genes are both being expressed at the same time. How would that end up with any resolution or stability? That stability of the two states comes from the regulatory aspects of these two transcription factors. CI preferentially and cooperatively binds to DNA at OR1, right next to where the CRO genes are initiated for transcription. When it binds here, it blocks the ability of CRO to work. That is, CI can turn off CRO. It blocks it. Right? This already gives us some idea of how the lysogenic state might be stable. When the lysogenic state is active and CI is present, it actively blocks and turns off the genes for lysis. A very similar thing happens in the opposite direction. CRO preferentially and cooperatively binds to DNA at OR3, which is right next to where the lysogenic genes are transcribed. When it does that, it blocks the transcription of the lysogenic genes. So in a symmetric way, CRO turns off the genes responsible for lysogeny. That explains the stability of the lysogenic state, or sorry, the lytic state. It's also known that stress increases the degradation rate of CI. Right? So if CI is actively shutting down CRO and all of a sudden there's stress, well, stress increases the degradation of CI, it drops down, that can release the block on CRO and allow lysis to take over. So this explains a lot of the basic structure, explains a lot of the basic phenomena that we see in this cell-fate decision pathway. Summarized here, we have the lytic pathway, or the lysogenic pathway, includes expression of CI. CI preferentially binds to OR1. When it does this, it blocks CRO and the lytic genes. When the lytic genes are expressed, one of these is CRO. CRO preferentially binds to OR3 and blocks the lysogenic genes. So this is this cell-fate decision. Quite a simple but elegant mutual repressor system where CRO turns off CI, CI turns off CRO. This explains a bit about the stability of the system. And gives us a qualitative understanding of what's going on. In summary of what we've seen here, we've discussed how bacteriophage, viruses that affect bacteria, have these two life cycles. Lysis, producing many copies of the phage and destroying the host cell in the process. Lysogeny, in which the phage DNA integrates into the host genome and passively is inherited as the bacteria grows and divides. And underlying this decision pathway is this mutual inhibitory pair of transcription factors, CI and CRO. All right. This is a summary of the biology of this lysis lysogeny decision pathway. In the next video lecture, we are going to get into how can we build a model of this system, and then we can learn something about the regulatory structure from exploring that kind of a model. For now, be well, take care, and I will see you all soon.